So there's something fascinating happening inside your body that most people never think about. Your gut microbes might be influencing not just your digestion, but your energy balance, body fat, blood sugar, and even how your metabolism decides whether to store or burn fat. So today, we're breaking down 3 major ways the microbiome impacts metabolism, such as energy balance and body fat, blood glucose control, and insulin resistance. And we're going to start with energy balance. So, in what ways can the microbiome, fiber, and diet influence how much body fat we store, you might ask? To understand this, we need to go back nearly 20 years, one of the earliest microbiome experiments that put this whole field on the map. Researchers took the microbiome from an obese human and transplanted it into a mouse. The result was striking. That mouse became obese. That single experiment led to a whole wave of research, including one of the most compelling lines of evidence involving identical twins. Now, this is where things get really interesting, because identical twins share the exact same genetic code, so genetics can be removed as a variable entirely. So even though they share identical DNA, often grew up in the same household, ate very similar diets, had very similar environmental exposures. So, due to a study where founders showed something remarkable, identical twins can be discordant for weight, meaning one twin can be lean and the other can be obese. So the microbiome transplant mouse study, the approach researchers took was to isolate the microbiome effect. They transplanted gut microbiomes from each twin into germ-free mice. Mice raised in sterile conditions with no microbiome. One mouse received the obese twins microbiome, the other received the lean twins microbiome. Then both mice were fed the exact same diet, the exact same calories, no differences in intake whatsoever. And the result? Mouse with obese microbiome became obese. Mouse with a lean microbiome stayed lean. So same food, same calories, different body composition. Now, how microbiome affects energy balance. So there are 3 major mechanisms we need to focus on: satiety, how full we feel, fat metabolism and storage, and third, gut barrier function. So how does the microbiome influence how full someone feels after eating? And the answer, this connects directly to some of the most talked about drugs in medicine right now, GLP-1. GLP-1 agonists like Ozempic and Wegovy. These drugs mimic a natural gut hormone called GLP-1, short for glucagon-like peptide 1. There's also another key hormone, peptide YY, or PYY. When we eat dietary fiber, gut microbes ferment fiber, they produce short-chain fatty acids. These SFAs activate G-protein-coupled receptors in the colon, and this triggers release of GLP-1 and PYY. So the physiology is fiber, microbes, SCFAs, gut hormone release, and then last, satiety. Now here's the issue. About 95% of people are not getting enough fiber in their diet. So that likely means lower GLP-1, lower PYY, reduced satiety signaling. So as a result of lower GLP-1, that could also mean higher glucose or something to do with higher glucose levels in the blood. This could also mean rigid ECM, where the immune cells, they cannot get inside the tumor because of the thick cross-linking, which excludes the immune cells. So therefore, the immune cells cannot get inside the tumor or cannot penetrate the stiff, thick ECM as a result of low GLP-1. So GLP-1 does other things as well, such as slows stomach emptying, so food stays longer in the stomach, which can cause nausea, so reduces appetite, and increases insulin release from pancreas. So GLP-1 is a good treatment, or was an original diabetes treatment purpose. That's why GLP-1 drugs were first developed for diabetes. But since drugs are so expensive, it could be better to have foods that could stimulate GLP-1, like, for example, allulose, which is a rare sugar that acts differently compared to normal sugar. So now on to a food study. There was a controlled human study where participants ate 2 burgers. So first burger was pork plus cheeseburger, and then the other one was tofu burger. So they were matched for calories and macronutrients. The only difference was the fiber content. Tofu burger had significantly more fiber. So the results after eating was higher GLP-1 after tofu burger, higher PYY after tofu burger, and then greater satiety. People felt fuller, even though calories were identical. So fiber, not calories alone, shifted hormonal response and fullness. Also, the high fiber could also help with the microbiome interacting with the biofilms, possibly and theoretically reducing the chances of harmful biofilms forming. If you haven't seen my other biofilm videos, that biofilms can sequester nutrients and lead to nutrient deficiencies. So with the tofu burger would be better than the pork and cheeseburger. Now here's some benefits to short-chain fatty acids. Number 1 is activates brown fat, which is a metabolically active fat that burns energy. It can also be activated by cold exposure and short-chain fatty acids from fiber fermentation. But the problem with cold exposure is that it could possibly upregulate PGC1A way too high. That could actually reduce NK cell numbers. So might want to be careful with the cold exposure. So the other benefit of short-chain fatty acids is that it also reduces fat storage. So SCFAs block fatty acid uptake into fat cells, reducing new fat accumulation. Therefore, there is not interleukin-6 being secreted by the excessive fats or belly fat, because interleukin-6 can cause iron trapped in macrophages. If you haven't seen my that video yet that I've made already, feel free to check that one out. Now, what role does gut barrier function play in metabolism? The answer: the gut is lined by a single-cell epithelial barrier that controls what enters the bloodstream. One major concern is lipopolysaccharides, or LPS, also called bacterial endotoxin. So it comes from gram-negative bacteria like E. coli. So normally LPS stays inside of the gut. It is harmless in that location. But if the gut barrier is compromised, LPS enters the bloodstream and then triggers systemic inflammation. The consequences of LPS in the bloodstream: LPS causes whole body inflammation, measurable via CRP, or C-reactive protein, and increased resting heart rate and lower heart rate variability. Key affected organs: fat tissue, liver, pancreas, and muscle. These are critical for glucose metabolism and insulin sensitivity. In human observations, LPS-related dysfunction correlates with obesity, which means interleukin-6 possibly secreted, and then iron trapped in macrophages, and then iron deficiency, and then type 2 diabetes, and then non-alcoholic fatty liver disease. All show dysbiosis, imbalanced microbiome. So that means that LPS could negatively impact your immune system, because 70% of your immune system is in the gut and influenced by your microbiome, and also increased intestinal permeability. So when you connect all 3 systems together, satiety, fat metabolism, and gut barrier integrity, you see a unified system where the microbiome is deeply integrated into energy balance. Thanks for watching, and I'll see you in the next video.